So today, this week's topic is about spot and the surface. I would say it's it's like the basic, the fundamental of the spot and the surface. We are not going into the advanced part. Spot and the surface are two basic analysis modules in Imaris, which is quite important because in my experience, if you know how to use spot and the surface, you would probably be like already like media or a little bit like pro users already, because if you combine the spot and the surface, it can be used in a lot of applications already. And we have some new features also in the latest version of Imaris, which will let the spot and the surface become more powerful. So I will introduce it in my training sessions. Okay, so let's go. Oh, by the way, if you are new to these training sessions and don't know who I am, I'm Arsene from Taiwan. I'm sales and application specialist, cover Australia. If you have any questions about how to use Imaris or etc., so you can email me at ed.chang@bigplan.com. All right, so today's learning objectives is, before I talk about spot and surface, I think it would be helpful if I also mention the measurement points. Measurement points is one of the analysis tools in Imaris which can measure the distance between two or multiple points in 3D and the angle between the two lines. It's quite easy to use, so I think it would be helpful to include it in this basic spot and surface analysis. And second is how to use and when it's good to use a spot. And it will include how to create a spot automatically and do some manual edit, how to check the different statistic, how to filter the group or the desired ones you want from all of the results, and how to label them in different colors, and how to do the color coding based on the single color or based on the statistic. Also, how to export the statistic to Excel sheet or text file, and how to annotate statistic. So basically, in Spot, I will cover this topic, and this topic can also be applied. The steps or the concept can also be applied to other analysis modules in Imaris, including surface. So when we talk about surface, I will mainly focus on, also go through the creation wizard, but I will not go through all these topics again. I will more focus on the different surface material rendering and export to different format, including the file for the 3D printing. Last topic today, objective today will be the shortest distance calculation between spot and the surface object, which is a new feature in Imaris 9.5, which is the latest version. Okay. So I will just show you quickly about the analysis modules I'm going to walk through today. First one is the measurement points using the slide. So first one is measurement points. As you can see, measurement points allow you to quickly measure the distance and angle, etc. in 3D. Okay, so now I will change to Imaris and show you how to use measurement points in a quick way. So maybe I will use this one as an example. Okay, so for this plant cell demo image, let's say if you want to just quickly measure the distance between any two of these green cells, then it's probably good to use measurement points because it's really easy to use and it gives you a result in few seconds or few minutes, depends on your operation. So if you want to use the new measurement points, you can find it here on the object list. Above here, there is a shortcut, add new measurement points. So you just click on that icon and this object will be added into the object list. So here, Imaris gives you a brief instruction of how to add the measurement points. Basically, you have to switch your pointer to select mode, and you will see this cubic in 3D space. And by using the mouse scroll, zoom in, zoom out, you will be able to change the size of this cubic. This is where you want to place your first measurement point. Okay, so depends on the options you choose here. Imaris is able to detect different things to create the measurement points. So first is all visible channel. So if you have multiple channels, it is better to choose a specific channel based on the object you want to measure. So for this example, I want to measure the green ones, so I will change to green channel. So Imaris will sample the green channel intensity inside this cube, then try to put the measurement points at the center of the intensity based on the channel you select here. So it's better, if you have a specific channel, it's better to choose it here, specific channel. So here, for example, I want to measure distance between this one and the button. So here I just put my cube around the center and hold the shift and the left click. So now I have the first point here. Again, for the second point, I just put my cube around the second object and hold the shift, left click. And after you put the point in 3D space, it's better to check whether the position is correct, because sometimes in this view, you see the point here, but sometimes if there is another object above, then Imaris may use that object as the center. So it's better to rotate the sample and check. But immediately you can see there is a distance already shown on the screen. Okay. And if you go to intensity here, Imaris will also show you the intensity profile in 3D across along with this line in the 3D space. So this is the intensity profile along with this line in 3D. And if you want to measure the angles, you can go to the setting tab and check the angle. Then you have to put another measurement points to be able to measure the angle. So maybe here, go back to edit, and maybe I use this one, shift, left click. So now I have 3 points, and between these two lines, I have this angle displayed on the screen here, and also the distance. So that is how you can use measurement points to quickly measure the distance and the angle in 3D space. So if you notice, we have a lot of options here. So we can change the shape of the points, and also we can make the line mode in the polygon, which is now we are using. Polygon means we can make multiple lines. Hold the shift, left click, so make a polygon. If you want to measure different distance in like 9 pair, so you can change to the pairs. So now we have two lines and with distance. This is one way to make the different set of the measurements. Another way is to add another measurement points. So now you can have another measurements like this. So what's the benefit of using the 2 measurement points is you can color code them in different color for visualization. For example, one is red, one is yellow or orange, like this. Okay, and all the measurement results can be found in Statistic tab here. Let me just move this to here. So you can see if I go to, when I go to Statistic tab, you can see the distance sum, and if you go to detail, you can see the statistic for each pair. So distance A, B, C, D, and for measurement point 2 is A, B only. And also the intensity center and position like this. Okay. So I didn't see the angles, so maybe here, oh, because I didn't use. So if I check this angle polygon, I may be able to see the angles as well. Yeah, angle here. Okay. So to export this result in the Statistic tab, you can use this floppy disk shortcut here. So you notice there is a single floppy disk and a multiple floppy disk, which the single floppy disk one is to export only the current viewing statistic, while the multiple floppy disk one is to export all the statistic here into a text file or Excel sheet. For the format, we can export as the CSV file or more useful XLS file or XML file. Okay, to the location you want to export. So that is how you can export the statistic here. So basically, that is how we can use the measurement points. Okay, I will go to the next topic. So next topic will be is the spot and the surface. So here, as you can see, in the previous session, we talked mainly about the visualization of your image. Now we will go into the part which is how to analyze, get the data from your images, which we have to go through a step called the detection or segmentation, which is to separate the target object from your background or from the other components. So in Imaris, we provided different tools for different applications for the segmentation of the different applications. The basic two are spot and the services, which is also very powerful. And the more advanced ones for specific applications are cells and the filaments, which hopefully we will talk about in the later or future training sessions. Today we focus on spot and the surface and the basic part. So what can spots do? From this image, I want to give you a brief concept of what spot can do. So on the left is a cell standing with some proteins maybe. And let's say if you want to count the numbers of these green proteins, then probably it is good to use spot. So in this example, as you can see on the right, after the spot is segmented or the protein is detected using the spot modules, you can see there are a lot of spots being created on the the image, which the position is the center of this green intensity from each individual standing, and then the numbers will be represented to the number of the green proteins like this. So if you want to count the numbers of this sphere shape objects, then the spot is a good tool you can use. And the spot is good to detect the position as well. So if you, for a different shape of object, it's maybe not the sphere, but if you just want to know the position information, maybe spot is also a useful tool for you. So numbers and the positions, and if your target object is a sphere shape, then the volume can also be measured using the spot. So most of case is the position and numbers. Sometimes the volume if your target object is sphere shape, and also intensity as well. Intensity. So after the spot is generated, you can measure the intensity of all channels inside this spot. Okay, so I will show you in Imaris. So here is just a summary of the Spa statistic, including the number, count, position, and distance between individual spots, or the spot and other objects like surface, and diameter, intensity, etc. Okay, so now I'm going to show you how to use the spot in Imaris. So for example, this one maybe. Okay, for this cell image, let's say if I want to calculate the number of these red kinetochores, for example, the number of these red structures is probably useful. Spot is probably useful. So I will show you how to do it. To create a spot here, like measurement points, you just add a new spot from this shortcut here. So once the analysis object like spot added, Imaris will show you this creation, we call the creation wizard, which is a step-by-step based process. You just follow the instructions of this menu, then you will be able to get the automatic spot creation result. So I will walk through this wizard with you. So first, you will notice there is a skip option here. So if you like to start from the manual creation, you can just skip automatic creation here. But today we are going through this creation wizard. So here, you can see there is a February creation parameters, which I don't have any here, but after you create a spot for one of your images, then you can save that creation parameters into this drop-down list. So in other similar images, if you want to reuse those parameters, you can just find it from this drop-down list here. Okay, so now we don't have anything, so I will just use default. And we have the ability to detect the spot just using the region of interest, not the whole images. So if you want to detect only the region of interest, you can check this option. And I will quickly show you, if you check this option, you go to next step. This step, you can choose the region of interest you want to measure or detect the spot. Sometimes you just want to detect in a specific small region of interest, then not the whole image, then you can use this option. Check this option and go next. And by adjusting these arrows, you will be able to change the region of interest. like this. Just remember to change your mouse pointer to a select mode in order to drag these arrows to change the region of interest, like this. So if I use region of interest, Imaris will only detect the spot inside this region of interest. And then you can actually do a multiple region of interest as well, like this. Okay. Right, so for the training purpose, I will just use the whole image. Okay. So second option is the different spot sizes. So sometimes you don't care about the size of spot, you just want to detect the position. So you may not need to check this option, but if your object is a sphere shape and you want to also measure the size, then we can check this option as well. So the spot Imaris created will be different sizes. So you will notice if I check this option, the step will increase. So if I didn't, it's total 3 steps. But if I check this option, it will be 5 steps. Right, so I will just show you without the different spot sizes. Then we will come back later. And third option is the feature I mentioned, shortest distance calculation. For now, we will just leave it. So as you can see, total 3 steps. So second step, decide a source channel. So here we use the red channel. This is quite important to make sure you select the correct channels to detect. You want to detect. So here I use red. And the estimated XY diameter, which is the diameter of this target object. So how to measure it? Usually you have to go to slice mode and measure this size. For example, here, left click and the left click, then you can see the size here. So here, if you have some large size object or small size object, here we recommend you to measure the medium-sized ones. So here you see there are some large ones and some small ones, but here I try to use the middle ones, medium sized. So about 0.47. So here I will just put 0.47. No, it's sorry, yeah, 0.47, like this. Okay, it doesn't need to be very precise. Usually when you first test on your image, you need to test the multiple numbers in order to get the best ones for your image data. Then after you find the good ones, you can try to reuse it for the other similar images. So here I start from 0.47. And we have this model PSF elongation options. This is for the correction of the Z elongation. As you can see from this image, also it's quite obvious we have the Z elongation, and usually you will see this elongation on your images. So if you want to correct this elongation in order to have a better position and also better size information, then we can check these options. So I will show you the difference. If I didn't check it, you can see the spot I created is all a sphere shape, like this. And that is why we will miss some informations because of this elongation. So if I check these options, I need to give Imaris a diameter. You can start from the 2 times of your XY diameters, and usually that is the Z resolution and the XY resolution. Z resolution is usually double XY resolution, but depends on the real situation. You may want to change the number as well. So let's see how it goes. So based on the default setting, it looks not bad, but if you want to increase or decrease, you can change the number here anytime you want. 1.2. So it will be longer or shorter, depends on these numbers you input. So it depends on the real image, you can try to modify these numbers. And the third option, another option here is background subtraction. So usually for life science sample, we usually check this option because the background is not, and the signal and the background is not very consistent. Usually there is a bright part and a dim part, and the background is not even. So if you check this option, Imaris will have a better detection of the spots. Okay, so let me go next. Third step is to filter or to remove or increase the spot detection. You can think like sensitivity. So to increase the sensitivity, Imaris will be able to detect more spots on the image, but there is a risk of detecting also the background. So it depends on the image, you can find optimize the threshold for your image like this. Okay, this is quality filter, and quality is basically the center intensity. It's based on quality calculation is based on the pixel intensity at the center of the spot. So if I use the lower threshold, it is able to detect the dimmer or low intensity spot, but sometimes it is background. So you may want to adjust this threshold properly. Okay, so now finish. So if I just detect the number of the spot, only 3 steps, I can get a result like this. And where to find the numbers, I can go to statistic, and overall, now I can see the number of spots, total is 64. Okay, like this. So if I want to use the different size, I can recreate this, rebuild this spot creation from the second tab, creation. So once the spot detection is complete, you can always go to the creation tab and try to rebuild from middle step, like here, spot, or from the radial. So here is also the place where you can store your parameters for batch. So if you store your parameters here, let's call spot 1, and you check the location here. Already exists, yes. So next time when you create a spot, you will be able to reuse these parameters here. Okay. Yes. Okay, so let's rebuild from the beginning. Here I want to check the different spot sizes. So I use the same parameter, and we have extra step for the spot size. So here we have to detect the spot region first, and usually for the biological samples, we also use the local contrast instead of the absolute intensity to be able to have a better detection. So what is the spot region? Here, spot region is for calculation of the spot size. So here, let me change the display from the also slice view to the ISO surface view like this. Okay, so the gray area you see on the screen is the spot region, and this threshold is to change the detection of this region. And the detection is based on this algorithm, either the local contrast or absolute intensity. Okay. So here you want to make sure the gray area covers the shape of your object. Like this. So most of the object is covered by the spot regions, and we can calculate the diameter of the spot based on the region volume or region border. So here in Imaris, if you are not sure about the meaning of some options, you can put your mouse or pointer on these options and right click, show help. Then usually Imaris will direct you to the menu for these functions. So here, as you can see, we have the region volume and the region border, which is for the region volume. For the region volume, the volume of this gray area or region will be used to calculate the diameter of the spot. And for the region border, Imaris will use the shortest distance between the center to the edge or to the border as the diameter of the spot. So if you want to measure the volume in a more precise way, maybe the region volume is good for you. For the other cases, maybe region border will looks prettier. So I will show you the different result. So region border, you can see there are some very small spot on the screen. Maybe it's because my region is not big enough. And for the region volume, when I use the same threshold, you can see there are some difference between the two results. So I think the most important thing is you find a good one for your analysis. Then you, for the other images, you continuously using the same algorithm in order to have the consistent result. Okay, so for the spot, we can have a different display as well in the settings. And if you want to manually add or delete the spot, we can go to this edit tab. And here you can change your mouse to select mode and the left click to select specific spot and click delete to remove the spot. If you want to do the multiple selection, you can hold the control key and the multiple select, like the Windows. Hold the control key and the multiple selection, or you can use these different tools, selection tools, for the multiple selection, circle or box. Okay, and also we can add the spot manually. Here we also use the specific channel, and it's like a measurement point. You can see this cube, and use the mouse, scroll, zoom in, zoom out to change the size. Place your pointer to where you want to create the spot and hold a shift, left click. Then you can create a spot based on the pointer position and the channels here. But the manually create a spot, if you want to create also this elongation shape, you have to manually change the Z diameter here. Okay. Right, so all this edit, the number will be reflected in real time in this statistic here. So here we are looking at the overall, which shows you the numbers, but if you go to details, we have more. So for example, the diameter and the distance from origin, position, also intensity here. So there are a lot of statistics you can measure by using the spot. Usually there is more in this drop-down list, which you can configure from the preferences. So under the statistic, if you go to this gear icon, tool icon here, Imaris will open the preferences for the statistic. Then you can check or uncheck the statistic you want or you don't want. So here, usually we recommend just check the ones you need in order to save time for the PC calculation. So here you can see a lot more for SPOT as well as other analysis modules. And also, if you right click on this statistic and show help, Imaris will lead you to this statistic reference. And under the SPOT, for example, it will show you how this statistic is calculated. And it will show you the formula. So if you need this as a reference, or you are not sure about how it is, whether this is the right one for you to use, you can always go to here and check the definition of each statistic. Okay, so here, and for the export, it's the same as measurement points. You can export the current statistic by using this single floppy disk icon, or if you want to export all the statistic into one file, or at one time, you can use this multiple floppy disk icon to export all statistic here. So here is the details, and if I select one of them or multiple of them, you will see it highlighted here in the table. And if you just want to show the one you selected, the result you selected, you can go to the selection. So here is only the one you selected. And also here, you can duplicate the selection to new spot. So it will clean up your result. All right. So you can select the ones you want by manual selection, or if you go to the filter tab, here you can add the statistic filters to filter out the desired object, for example, based on the statistic. So for example, if I want to filter out the large ones, so the volume, I can use this threshold to filter out the large ones. So here I use 0.233. So for the spot larger than 0.233 micrometer cubic, it will be selected. And here you can duplicate the selection to new spots. Or if you want to delete the small ones, we can use the upper limit here and select the small ones, and here the edit, we can delete them. All right, so either way you can do, and not only one statistic, you can also add multiple statistic in this filter tab. Somehow it's freezed, so I may need to restart. All right, sometimes this happens. All right, so let me share the screen again. I guess now it's disappeared. All right, so let's go back. Let's go back to this one. All right, so here let me just reuse this again. And I click this green arrow to quick finish. So now I have the same result. All right, so let's continue. And filters, here we can add multiple filters as well. So if you want to filter based on the shape, based on the intensity, you can add here. Okay, and also, if you want to annotate this result on the screen, for example, the volume of this spot I want to show on the screen, I can change to the volume display and select the spot and use this button here, add annotation from selection. So now you can see the volume being displayed on the screen here. And there are a lot of spots, so it's not easy to see. So usually you will have to duplicate this spot and go to here. You can see just this one is more clear. And from the right side here, you will notice there is an annotation panel. And if you go to this one we just created, you will be able to add other annotation for other statistics as well here. That is how you can add statistic annotation here for the snapshot or for the animation. Okay, so here, if you want to delete, you just click delete. Yes, so now you will delete it. Okay, so that is the annotation. And for the color tab, you can change the color based on the best color, or you can change the color based on the statistic. So here, if I use this one, so if I want to, let's say I want to color coded my spot based on the size. Oh, here I use all the same size. So let me just try to quickly create the different size of spots. Okay, let's say this one. Okay, so now I have different size. Then let's say if I want to color coded my spot based on the size, I can choose volume. So now the large ones will be color coded to the red, and the small ones will be color coded into the purple or the bluish, like this. So you can have a visualize the statistic using the color code. So here I have all the statistic I can use, and if you notice there is some orange icon here, which means these are the same. There's no difference. So there is no meaning to do the color coding for this statistic, but for the others, it will be different. Okay, so one of the useful applications using the spot I will introduce briefly is using the intensity. So as you can see, we have also the other color, white color on the screen, which is another protein or another component. If I want to measure how many kinetochores also express or overlapping or colocalize with this dynectin in the white color, one of the quick way is to create a spot based on one of these components. For example, I use red channels, and by using these filters, I can add intensity, maybe mean for channel 4, which is my dynectin channel. And by using this threshold, I can find which kinetochores are also include high dynectin intensity, which means there is also the another components overlapping in the same 3D space. So for these ones, I can duplicate. So now I have these ones with the colocalized between the kinetochores and the dynectin. There is just a quick example of how you can use spot. But I think in the future trainings, in the later sessions, we will talk about more advanced applications for the spots. Just quickly show you how it can be of use. All right, so that is the basic of the spot creation. I hope I have covered most of the things. Another is labels. So here, once you filter out this target object, if you don't want to create too many separate individual spots or object on the list, sometimes you can just label them in different color. So I want to show you how to do it. It is for you to categorize your results by using the label. So if you go to this camera label tab, and here, first you will have to add a label group. So first create a label group using this button, and maybe this is for IMB, UQ, and enter. So now I have this new group of labels, and now I can add labels. So add a new label by right click and add a new label to current group. So this one maybe I say colloq once, and I can change the color to maybe, let's say green. Okay. So now I can, because I already have some target object selected, so I can right click. Once the label added, the select ones will be tagged to this label automatically. So we can right click, remove, or add label to selected object. So I can say this is colloquial ones, and the opposite. For the opposites, maybe I can use another ones. Colloq. So maybe I change to some purple color. Okay. So now I have two labels for my spot. So in the color tab, I can display the label. Label override the base color. So now I have two label groups. So now I have two colors. And also in the statistic, you can see there is another column, which is the name of your label group. And if the object is being labeled, you will see the label also when you export the result. So that is a way for you to sort out your statistic for the target object as well. So that is how to use the label in Imaris. So I think it's time to go to the surface. So for the surface, as you can see, the shape, or I think it's the shape that surface created is much more flexible than the spot. So if you want to measure the volume of some irregular shape, like some organs or some components, like this example is a mouse embryo, so the brain size, liver, or some intestine or spines, this kind of irregular shape, more complicated shape ones, then you may need to use the surface to measure the precise volume or to render the shape. In surface, we have automatic or manual creation. Today, I will focus on automatic ones. For the manual ones, we will talk about it in the later training session. So for example, this one is a Drosophila brain, which we can use the surface to render this brain, and we can measure the morphological statistic, including the volume and the longest axis, shortest axis, sphericity, surface area, etc., from the surface statistic, which we cannot use the spot to get this result because of the shape. Okay, so for the surface statistic, it's similar with spot, but including also the morphological statistic as well. Okay, so now I'm going to quickly show you how to create the surface. Here, maybe I will just use this one. Okay, this is a blood vessel. And let's say if I want to measure the size, the volume of this blood vessel, apparently I cannot use the spot to get this result. So I have to use surface. So here, next to spot is surface. This creation wizard is very similar with the spot, so I will just quickly go through. So go to next and the desired channel. In the surface, we have the smooth option. So here, usually we use smoothing and use the default number to see if this surface details can be satisfied. This smooth option, if I uncheck it, it will show you show you more details on the surface because there is no smooth out of the small pixels. So usually for the better visualization, we will check this smoothing unless the object you want to see is very small. So the default number here is the 2 times of the voxel size. So here, voxel size is 0.16. So this default is 0.32, which means the surface smaller than 2 voxels will be smoothed out. Usually we check this option. And for the thresholding, if the object is quite simple, for example, this single individual big structure, I can use absolute intensity so to easily get this structure. But if there are a lot of components, or for example, cells, a lot of cells in the image and the background is not so even, in that case, we have to use the background subtraction for a better thresholding. And for the diameter here, you just give Imaris the largest sphere which fit into the object. For example, if you have a multiple cell, you want to measure the size of the cell and put it here. Put it here. So here I just use the absolute intensity. Maybe I can show you the difference. This is absolute intensity, and I can see some small object here. But if I go quickly measure here, the size is about 3. So if I go to here and put 3, there are some difference. So there are better segmentations based on the intensity size you put here. But here, mostly I want to render the complete structure, so I don't want to give too much details. So I just use absolute intensity and try to adjust this threshold to have more complete structure like this. So if you want to close this hole, sometimes you can just increase the threshold, or sometimes you can increase these surface details to have a more general surface creation like this. All right, so here we have the option to split touching object, which is for a more complicated detection, which maybe I can show you if we have time today, but if not, then in the advanced surface creation, we will try to show you. This is basically for Imaris to automatically split the touching object using the watershed algorithm. But here we only have one object, so we don't need to use it. In that last step, we can filter out the small unwanted surface using this threshold. Okay, so you see the small ones has been filtered out. So finish. So this is the surface we created. And here you can see the same tab spot. You can store the parameter for batch. And this tab is for the draw. This is a manual surface creation, which we will talk in the later training sessions. We will focus on this, how to manually draw a surface. This is quite useful in some cases which the object is not so easily detected using the automatic algorithm. And the edit tab, where we can cut the surface and do some masking, which is also the topic for the next session. But here I want to quickly show you how to cut the surface just in case you need it. So here, if you want to separate the surface, for example, this is not actually the one single surface, but the two, and it should be separated from here, from the middle, then I can use this cut surface. So here, I just quickly show you in this tab, I change my mouse to select mode. Let's say I want to cut here. So I hold the shift and the left click. So there is a cutting line here. So just to remind a quick note that the cutting line in Imaris is always a vertical. So it's always a vertical line. So if you want to cut here, cut this part, you have to rotate the object in the correct orientation in order for Imaris to make the cutting line. Okay, so you will have to rotate your object, right? So let's say I want to cut here, so I just cut surface. So now it became 2/2, right? So that is how you can cut the surface using this function here. And the filter is same, and the statistic, oops, again. All right, so let me just quickly come back. All right, you can still see my screen, right? Okay, so let's go to here. So let's say, okay, I have this surface I just created. Then in the statistic, you can see the same layout as the spot, which we already show you. So I won't go through in the detail, but here under the preferences, you will see a lot more statistic, including some morphological statistic like bounding box. So bounding box sometimes is useful if you want to measure the longest axis or shortest axis of the object to compare the growth or compare the size. So this is bounding box, as well as the sphericity or ellipticity, etc. So you can check here and check the ones you need to include in the statistic. Then you can do the filter or the color code as well here. So the difference between the surface rendering and the spot is here, the materials. For surface, we have different materials. So now the default one is these shiny ones, but you can change to a different material. So different texture according to your sample. One of the useful ones are these transparent ones. So if you want to show the shape of this structure as well as something inside, then we can try to use these transparent materials and to visualize something inside if there is any. Okay, so you can change the materials as well here. See, okay, surface materials, and once the surface is created like this, some user may want to 3D print this structure, right? So how to export this for 3D printing? You can go to 3D view here and export selected object here. And Imaris can export this as the via Emerald2 files or the IB files. For the other files, maybe you have to export to these one of these two files and file type format and convert it using the other software. For now, we support these two formats. Right, so that is the surface. Just a little heads up of the function for the mask. So mask function I can also introduce. I think we still have a little time. So for example, for this case here, for our nuclear staining, we have the nuclear structure here and other some background noise here. If we want to clean up the background, we may be able to use the mask function to clean up this background. So here, let me quickly show you. I create the surface based on my blue channel, and I use absolute intensity. Okay, so here, basically I find a good surface for my nuclear structure, and I use the filter to filter out the small ones. So once this surface is created, in the edit tab, you can find this mask. So if I mask, the channel I want to clean up is blue channel. So here I select the blue. So I tell Imaris I want to set voxels outside the surface to 0. So every blue voxels outside this surface will be set to intensity 0, right? And there is option to set inside surface to whatever. So that's the opposite way. But here I want to remove the background, so I click OK. So you see now there is a new mask channel here, which if I disable the original channel, now the background outside this nucleus are being removed because of this mask function. So this mask is quite useful to separate some different structures, and sometimes you need to combine with the manual surface drawing in order to separate the different region by using the mask, which we will introduce in later training sessions. If you are interested, you must make sure you join that once. Maybe the name will be Advanced Surface Applications, something like that. Okay, so here, the last topic I want to cover is the shortest distance between spot and the surface object. So I may use this one, for example. Okay, so for this image is a blood vessel with some, let's say, cells. So if you want to measure the distance between the blood vessels and each individual cells in the green staining, how to do it? So in Imaris, we have this shortest distance calculation for you to quickly run this kind of statistic for these applications. So here the surface is created based on the blood vessels, very obvious. And we use the spot to detect the position of the green cells, right? So during the creation, during the spot and the surface creation, for example here, if you want to measure the shortest distance, you must check this option. You must check this option in the surface or the spot creation here, right? So if you didn't check this option, you can also check it under the edit here. After the creation, you can check the shortest distance calculation here. So before you check this option, if you didn't, there will be no shortest distance calculation. Because it takes more time to calculate this shortest distance based on the complexity of the image. So we didn't put it as the default option. So its default is you can have it unchecked in order to facilitate the process. So here for the spot, so shortest distance to spot, shortest distance to surface, I already checked. For the surface, I also need to check shortest distance to spot to surface. Okay, so it depends on what you want to measure. If for this one, we want to measure the shortest distance between each cell to the surface of blood vessels, then probably we don't need to calculate the surface shortest distance here. We just enable the spot shortest distance that is enough. So here, shortest distance calculation, enable. Let's go to here. Maybe I also need both for showing the spot calculation here. So once I enable the shortest distance calculation. So maybe you have to enable the both object you want to measure. So here I can find the shortest distance to surface. Surface is surface 1. So here is surface 1. For this name of the object, you can double click and rename it. So you will be able to see the different names here. If you have many objects and it's a bit confusing, you want to make sure you name it correctly so you can recognize it easily. Okay, so for the shortest distance here, this represents the shortest distance between the center of this spot to the surface of this red channel here. So we can use this filter function to easily filter out. So for example, I want to calculate how many cells are actually inside or in touch with the blood vessels. I can add the shortest distance and I turn off the minimum is -6, which means inside this surface object. I can turn on, I can say 0, turn on the upper limit and say 0, which means in touch, attached to the surface. So I can duplicate. Okay, so these green ones, or these duplicate ones, are actually the ones I want, which is inside or in touch with the blood vessels. So to be able to display it better, I can go to surface and I can go to the material. I turn it to change to the transparent one. So you can have a better visualization like here, yeah, all the ones we want. Okay, so if you want to calculate the numbers of the total spots, total cell, it's here. And the ones in touch or inside the blood vessel, the number is here. Okay, so we have two numbers. I want to show you how to group them. So if you want to export these two results together, you can add new group and put these two objects into this group. So if you go to the group, the object properties here, you will see the result for all the objects in the group here. So now you can export this in the same table, and then this name, this object, you can rename this object by your preferences. So it won't have the same similar names or very complicated names here. You can try to, this is all, this is touch, whatever. So here, you can try to change it and export it to Excel sheet. So it can be used in many different applications, including immunology, to find the granules which is inside or outside the cells, or you can use it on the organelle distribution inside the cell. For example, the nucleus, you use the surface, or for the organelles, you use in the spots. And you can use this shortest distance calculation to measure the distribution of the organelle inside the cell and color coded. So this color coded, you can do it here. So if you color coded this based on the distance to surface, then you will see the inside or in touch is the bluish color, and the far away ones are the warm color, like red or orange color. So you can color code it as well and take a snapshot or make the animation. So here there are a lot more applications you can use by using this native distance measurement. So I think it's really useful and a very good feature in the latest version of Imaris. Okay, so that is the topic we will cover today.